So hi everyone, my name is Duong, and I would like to present the oxytocin receptor today. And so in my project, I would like to first talk about the breakup oxytocin receptor. So on the right should be the protein tree structure of the oxytocin receptor of human, and the oxytocin receptor is part class A family 7 transmembrane Z protein couple receptor. And first, I would like to talk about the what we know about the oxytocin. Oxytocin is the hormone released by the hypothalamus and released from the posterior pituitary gland. And oxytocin have crucial function in sexual reproduction of both male and female in many species. Also in female, oxytocin plays an important role in uterus during childbirth and lactation. And here the oxytocin structure. And I would like to show on a step to find all the oxytocin conserved protein sequence. So first we go to ncbi.nlm.nih.gov, and on the search bar, the default should be all that database. We want to click on that, scroll down the drop-down menu, and choose protein because we want to narrow the search to protein only. And in the search bar, we enter oxytocin receptor and hit search. And when you hit search, you want to narrow the result of your search by the species on the left. We choose animal and the sources database with two rep6. And we hit on the first result, the oxytocin receptor of Homo sapien. And when we hit in the result, we can see on the right under analyze the sequence, choose identify conserve with domain. And then we going to hit on the specific hit search bar to link to the conserved protein domain up. And then on the link on the left, we choose specific protein. And we can use taxonomic group 3 to search for specific organism, or we just choose any organism in the result because they are on the oxytocin receptor of many species. And for example, over here, I want to find brown rats, so I choose rodent, and then hit the second result to go to the oxytocin receptor of brown rat. And the third one, I want to talk about 7 transmembrane Z protein couple receptor superfamily. So here the 3D structure of that particular superfamily. And based on the sequence similarity, the oxytocin receptor belong to rhodopsin type 1 Z protein couple receptor superfamily. So here the 3D protein structure of that. And the family is known for its function of transmitting psychological signal from outside of the cell to the inside of the cell through the Z protein. So the activate Z protein then bind and active numerous downstream effector protein that will generate second message in the psychological processes. And here's some accession ID of some species that I'm going to put in the MSA. So here human, here cattle, brown rat, house mouse, red shingleful, crab eating macaque, pig, sheep, zebrafish, Chamberis medaka, and here the multiple sequence alignment of the FATSA file of the previous species. So we can tell that in the multiple sequence alignment, of the oxytocin receptor domain from column 40 to column 340. The sequence are almost identical, maybe not identical, but these amino acids still conserve it due to similar characteristic. And the two reasons different in the first, the beginning and the end. So from column number 2 to 40, and from 300 to the end, 340 to the end. And there are high similarity in amino acid sequence for big cattle, sheep, brown rat, house mouse, human, and crab-eating macaque. Since these animals are mammal. And for zebrafish and Japanese Medaka, they also have high similarity for the two reasons, because they're bony fish. And in case of red shinglefro is the only bird, so the reason is different from the other species reason. And for phylogenetic tree, we can see that zebrafish and Japanese Medaka are both bony fish, so they form cluster together. The red shinglefro is only bird, so it stay by itself, and other species are mammals, so they form cluster together. So house mouse and brown rat are rodent, they form cluster together. Pig, cattle, and sheep, even though unrelated cluster, and the human and crab-eating macaque are both primates, so they close together. And here the percent identity matrix and the real diagram of phylogenetic tree. And we can tell that these evidence support the evolution. So for the motif sequence logo, the lower entropy, the higher certainty. So we focus in the total character would have the higher certainty to form the conserved protein sequence. And here, based on here's the sequence alignment of the conserved protein sequence that from the NCBI website. And from those information, I construct the conserved protein sequence. Start with ME, and here from the amino acid 40 through 340. And here the works cited for the NCBI website. And thank you for listening. That would be the end of presentation. Thank you.